Kia ora koutou, ko Barbara toku ingoa. I'm Barbara Anderson and I'm here with the Ahi Pepe project, and what we're doing here is this is our big experiment day. So here we're setting out the experiment. We've got the amazing bushy park here, and you can hear all the birds, you can see all the forest. We're down here in the newly restored wetland area. And what we're doing is we're setting out these traps so that we can see what the moth community is doing inside Bushy Park and the effect that the predator-free fence is having on the moth community, and how different that is from those poor communities that are left outside of the predator-free fence. So here we have the trap. Next to the trap, this is the moth trap with the vane and this light, and here is a little solar sensor, so when it gets dark, this will go and the light will come on, and it's a beautiful blue, white, glowy light, and that's really bright to a moth because they have different eye structure to ours. And so the moths hopefully will come into our trap and they will just fly in here, and in the bottom, if you can just see in here, there's a net, and then inside the net there's a whole lot of egg cartons. Oh, sorry, I'm standing up. And the egg cartons are all rough, like the bark of a tree, and so the moths, they'll fly in, they'll be attracted to the light because they think it's the moon, but then they'll go down and they'll snuggle into those egg cartons, and they'll be all nice and safe there, and they'll be quite alive and unharmed and happy there all night. So they'll just snuggle down as if it was the bark of a tree, and they'll stay there, and then when it's cool in the morning before it gets really hot, we'll come and we'll take the light off and we'll close up the bag, and then we won't kill the moths, we'll just take the moths and we'll put them in a fridge where it's nice and cool, and that will give us time so that we can take each net out and we can look at the moths that are there, and we can see what moths are there, and hopefully we'll be able to identify them. We may not be able to identify all of the moths because sometimes unfortunately you do need to kill the moths in order to be able to tell which species they are. But for this exercise, what we're actually going to do is just hopefully, because we've got the New Zealand expert taxonomist here with us on the camp, Dr. Robert from Manaki Whenua, we'll be able to identify most of the moths, and we'll count the numbers that we've got, and then we'll bring the moths back here to Bushy Park, and we'll release them back into the environment, and hopefully they will carry on. So we've got the trap here, this is our moth trap, and we've also set out these little black tracking tunnels over here, which are to see what's around that might be eating the moths. So of course there's the native birds, but there's also the introduced mammals. Obviously inside Bushy Park we're hoping and fairly confident that there are no kiori and no hedgehogs, no stoats, no possums. There might be a few mice, and the other thing there might be is possibly some native gecko or some skink, or we had one time in Orokonui Eco Sanctuary, we actually had a kiwi bill mark inside the tracking tunnel, with the peanut butter was eaten by the kiwi. And the other thing we've got is we've set up some little data loggers. So the data loggers are, and we might go and see them afterwards, they're just little kestrels, just little yellow ones that measure the temperature and the humidity and the wind speed and direction. And we'll have one of those at each of the sites. So we've got 3 sites here inside Bushy Park, this wetland area, a regenerating forest area, and the old growth forest. And then outside of Bushy Park we've got the same 3 habitat types, which are our controls, but they're outside of the park. So there's a wetland, a forest, and a regenerating forest. And that's the basis of our experiment. It's quite simple. We've got a question: what is the impact of the predator-free fence? We've got a treatment, which is the fence itself. We've got a control, which is the sites that are outside the fence. We've got the response, which is counting the moths. And we've got the replication, which is one trap here and one over there and one over there, and then a 4th one over there. So that's our 5 parts of the experiment, and that's how we're working with the Ahi Pepe MothNet.